I get it.
No, you won't.
I won't.
Anyway.
All right. And if you have questions, I think as you go, you're welcome to ask, right?
Yes. Yeah. Go. I mean, no, the thing is, if someone's has a question from the other site, I don't know what I'm not going to do.
Yeah, they'll just start yelling out. If they got questions from the other sites, they just yell out.
All right. Okay. Thank you. Thank you again, Mike, for the great introduction. I'm a jack of all trades and master of a few, but what's been really good about my whole time in industry prepared me for teaching. And so the big thing, what I'm going to talk to now, because I don't know how much you people know about receptors and enzymes, but when I first was a medicinal chemist, I worked a lot in the CNS, and the point is, and I also worked a lot in the when I did process work, we had a lot of CNS drugs. So to me, it was very good. And what I'm going to really tell you, you're going to see me walking around and holding model kits. And the idea is for models, that's how you can visualize things. I mean, I don't know how you all taken organic, right? And I don't know about if you tried to learn stereochemistry, if you didn't use it, if you didn't use a model, you were lost. And even after you're trying to do some of the synthesis works and you're having a problem, you don't grab your model kit, which I never understand why. I mean, who doesn't like playing with Legos? I mean, so the thing is, if you look at the model, you can actually visualize. And the people that were there yesterday noticed the Trovan structure. If you look at that side chain, you don't necessarily see how they overlap. And the cool thing about this area is the targets. And basically, I'm going to show you how they're so the same. Oh, did I turn it on? Oh, don't. Oh, come on. Oh. All right.
Do we mouse for that or arrow instead? Okay. Sorry about that.
Oh, okay. All right. Anyway, so the idea is with neurotransmitters, the idea everybody's heard of the lock and key, right? Of receptors. Nod, you can nod your head, say yeah. So the thing is, so the idea is there are a couple of ways. The most important is, and I found this area, and I have another idea, but I don't want for lack of time. So the hormone or the neurotransmitter plugs in nice with the key, turns the lock, everything's happy. The agonist will like when you're trying to get into, you forgot like using the hairpin. I don't think they do that anymore. And so you go in, clink, 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 and the agonist sort of gets in the lock, but doesn't always do the thing. Sometimes it can be stronger than the natural natural transmitter or hormone, or like it, or an antagonist which gets in there and does damage kind of thing. So anyway, so neurotransmitters, you all know that. I mean, all your biology people know they're transmitters. Of course, I'm not going to read all this thing kind of thing, but the point of that is it's really important for anything our body does kind of thing. And the point is, and what you're going to take a look at, you're going to see that they're very interestingly related. Quick cartoon, how they work. The neurotransmitters are made, in the vesicles. The vesicles go down into the vesicles go down into the transporters, and then they push them out into the cleft, go into the receptor, stimulates the signal, and then you get the nerve to go down, which is a good thing in the bag. Now, I want a quick look through here. What structural functions, what structural features do these guys have? Kind of thing. What functional groups do you all see that's important in these things?
Are you talking about the ones that are on the right side of the screen?
The left, on the right side, yeah. Can everybody see those? What commonalities do you see?
Yeah, what do you guys see?
Amines. We see hydroxide groups in everyone. Now over here. what do we have? What are the features? We have an amine,
Yeah,
right?
That's the indole ring that we see in tryptophan.
Right, but what are those? So what functional groups are the indole, the phenyl ring? What are they? They're what? They're the aromatics, right?
Aromatics.
And they're what kind of they are they? Lipophilic or hydrophilic? That medicinal chemistry is a horror hodgepodge because all the stuff you learned in all the other classes comes back to haunt you and haunt you.
Remember when the aromatics he's got there, they're all carbons and hydrogens. So are they polar or non-polar?
Yep.
Bunch of carbons and hydrogens.
Non-polar.
Yeah, they're
No,
non.
they're all
So are they gonna
non-polar.
like water or hate water?
They're gonna hate
Yeah, they're
more.
gonna hate water, so they're gonna be hydrophobic, the aromatics.
And the thing is too, they're all gonna have a similar, they're all gonna have that similar, they all have that nice amine to it. They end up, we're gonna have some chirality kind of thing, which, and all these things are gonna say, they're gonna have amines, they have hydroxys. So all of these things are part of what they look at kind of thing. And all the neurotransmitters, acetylcholine, serotonin, dopamine, and even some of like tryptophan, GABA aminobutyric acid, glycine, which is an amino acid, that's also a neurotransmitter. But if you notice, they all have these, most of them all have these ethylamine moieties kind of thing, So right here we have my favorite, dopamine, right? So dopamine has the hydroxy, the phenol groups, and that ethyl group. And what happens is here, the difference is they have maybe they have a methyl group here or something like that. So if you look at this molecule, you can do a lot of nifty things to change it to make it fix its function. So what he can do either increase the activity of a function or decrease the activity of the function. So take a look at this. Okay, so serine is what? What type of compound is serine?
Yeah, I just taught the students this on Tuesday. So it's an amino acid.
Amino acids, right? Okay, so serine, and you're going to see a motif coming through about amino acids in a slide or two. So serine, take serine, goes in, make to choline, and then N-methyl transferase. You'll hear about this transferases. Biochemistry is pretty good because if you want to do like methyl transfer means transferring a methyl. Decarboxylase means it's decarboxylated. It's not as bad as some of but it's really pretty good that they have pretty good names. And what happens is choline, they decarboxylase. From that, they make these methyls, and then acetylcholine, they stick an acetyl group on, and acetylcholine, and then remember, it gets transported to the vesicle. So, which is pretty simple kind of thing. So that's the first one. Then there's the biosynthesis of catecholamines. So catecholamines will come from L-tyrosine, which is again an amino acid. So we have the amino acid, and take a look at what's happening here. It goes into L-dopa, and L-dopa does what? What's L-dopa? What happened? How do we get from tyrosine hydrolase? What's happening? What's the reaction that's going on? Yeah, hydroxy. So we got now, and from tyrosine with the amine, we have that in the middle. So then L-aromatic amino acid decarboxylase does what? Decarboxylase. What I'm trying to explain to you that this isn't, if I remember can do some of this stuff, it's not that hard. You agree, Mike?
How do I answer that?
So anyway, so then what's the next thing? What do we have to do next is what?
Where are you at? You're looking at the L-dopa?
I'm looking at L-dopa, right?
Yeah, so we're going to dopamine. So what's the difference when you go?
So what happens to dopamine up to noradrenaline?
Oh, from dopamine to noradrenaline?
To L-noradrenaline.
Yeah.
What's the difference?
Adding a hydroxy.
Pardon? Yeah, hydroxylase. And what about this thinking? I'm going to use my cane. Give me my cane. Hell with my. So about what happens with this is,
Just don't knock the screen off the wall.
I'll tell you a story about this. I don't, so okay, so dopamine, and so what here is what kind of carbon is that? Yeah. What else is nifty about that carbon?
Chiral.
Cheater. Chiral. So L-dopamine, and then what's the next thing? What's the next step is putting a methyl group on the end. So if you go back to that, thing, that's where from that one amino acid, from tyrosine, we get all those neat amino acids. And then for the next one, another favorite molecule is serotonin. Instead of using tyrosine hydroxylase, they're going to use tryptophan hydroxylase. This time, instead, it's going to put one hydroxy on there. And it's going to do a 5-HTP decarboxylase. So I always found that this was very cool, that the majority of the neurotransmitters were made up, a lot of them were made up from amino acids. So somewhere nature decided, amino acids are so important for biosynthesis and proteins, but somehow they'd also got hydrolyzed of doing nerves and transfers of information electronically. I mean, I don't know, but you may not think that's cool, but I do. So, okay. So now we'll switch it and go into so you're eventually going to see all this prelogue is going to be very informative. So the next thing is opioid analgesics, and I bring that out especially. Sometimes I give talks that not to students, but to the lay people kind of thing. But also too is because, especially in where I live in a compound, in our complex, there are not a lot of scientists, or if there are scientists, they're not medicinal scientists. So the big thing is opioid analgesics have been around a long time. Opioid is meaning juice, they extract it, and the opioid is the natural or non-synthetic drug that binds to an opioid. An opioid is either, here's morphine, here's morphine, or one of the other ones. What's the one in the news that's a nasty drug that they're always worried about? Yeah, that's expensive.
I was gonna say Ozempic, but
No, that's number 2. So that's the first thing. So, I think you're going to see, if you'll see where I'm driving in the middle. So these are the two. Now take a look at these. What would you say if you looked at these two? Would you think they were the same? Right? No, that can't be. Just wait. So, history of opioids, and all of you that were subjected to my lecture yesterday, was that opioids have been around for a long time, kind of thing, and they used it, even in antiquity, kind of thing. And the big thing was back in the 1800s, they started using morphine with alcohol, which was called laudanum. And back then, though, the only way they didn't have injections, kind of thing. But they'd have no fear. The hypodermic needle was invented in 1856, and then people started having that kind of. Now, back before the Food and Drug Act of the early 1900s, when Teddy Roosevelt, what they used to call snake oil salesman, right? Have you ever heard of that? Snake oil salesman? And the big thing was laudanum here is laudanum, the universal household medicine, right? Those 10 to 25 drops, sedative, hypnotic, and a poison if used too much, kind of thing. So, but what happened was, there were a lot of people that were, in that kind of thing. And Queen Victoria, actually, everybody know about her. She was she was a nasty person, I think, to give, but she was addicted to laudanum along with a lot of other people. So here, okay, so now over here we have morphine, and everyone heard of the enkephalins, endorphins. Well, and who's gonna? I'm sorry, because I get tired. I want to ask questions. What are they exactly? What do you guys know about those?
About endorphins or?
And endorphins and enkephalins.
That's an interesting question. What do my students know about endorphins? I haven't taught them yet.
Well, no, they should. Don't they read? Don't you read? Okay, yeah, right.
When I think of endorphins, I think of like after you exercise, you have a higher amount of them.
You do, and so yeah. Yep, that's right. Okay, yeah.
It's more the body's endogenous type of.
Endogenous. There you got that. Yeah, you've got that thing. And it is the endogenous. So what happens is that's the methamphetamine. And when I was in graduate school, that's when Dr. Solomon at Yale, he was the one that, that actually discovered the natural endorphins. And that's how I landed my first job, because I knew about naturally occurring endorphins, which reminded me, I said, oh, that means, oh, you must be aware, you must be reading the literature. The only reason I knew it was one of my research advisors worked for. So, I mean, it was that kind of, again, doesn't matter who, kind of thing. And here it is sticking there. There is so it's a peptide, right? Everyone know what's a peptide? Peptide? You can shout it out. What's a peptide?
Yell it out, guys.
You can yell it out, yeah. We're all together in here. We might as well. Yes.
It's between amino acids.
Yeah, amino acids, and here it's tyrosine, glycine, methionine, and phenylalanine. So I wasn't going to build a peptide kind of thing. But so what happens is that will sit on there with no trouble. And okay, so this is the thing about, so the opioid neuron, which is not really, but an opioid is almost any neuron. So almost any neuron will be affected by morphine. But there are parts and what happens is the morphine receptors are there for pain and things like that. But the problem with some of these drugs, especially if you take them in high quality, it depends on the concentration of these molecules. So if you take a ton of drug, it will saturate. So even though it may have an affinity for the dopamine receptor, it might also affect the adrenaline receptor, beta, or may affect any other receptors kind of thing. And the reason, if you really take a look at the structure, those structures have stuff embedded in them. So what would happen is if you saturate something, so what they do now is they do assays. So the primary effects of the opioid, it says in reduction or inhibition of neurotransmitters, thereby it releases the thing into the cleft and shuts down the reuptake, and it just keeps stimulating the receptor. So. And if you think about it, that's how one of my students, it seems every year, every student wants to talk in my medicinal chemistry about street drugs, which, if you think about it, all of them have that interesting structure, which we'll go into later on. So opium, again, is a mixture of different opioids. And notice they all have what they have in common. What do they have in common? What do they have different?
3 rings.
Yeah, they have 3 rings. But notice they have 4 rings.
Yeah, they got that weird ring that juts out.
Yeah, they jut out. Yeah.
Yeah, a lot of these morphine derivatives too. There's Lortab. That's also morphine. Then we have like oxycodone.
But a lot of
And,
these, sorry.
And a lot of them are like, have this sort of parent structure in them with just slight variations. I sometimes jokingly call Lortab floor cab, because I imagine if you just scooped all the crap off the bottom of the floor of a cab and put it in a pill.
Anyway, but the thing is, so, and then, all this trouble they've been talking about of having people addicted to opioids. I will never understand how people can honestly think, looking at these structures, because if maybe what you want to so is sometimes look at some of the oxycodone and things like that, and you'll see that they have this thinking, they still have that nucleus, and they get it from the morphine. They don't. I mean, what they do is they used to do back in the din times. We used to try to make morphine. There's a couple of elegant systems to make morphine, but the plants, just like bacteria, make better drugs with penicillins. Same thing with this. So what happens is, codeine is also in there, and codeine is also, if you take, I don't know, they give codeine. Does people have codeine for cough syrup anymore?
Yeah.
Isn't that the best cough syrup? Yeah. It does too. Well, think about it. Think about it 150 years ago. People had a cough, and so how did you get better back in the day? You slept and rested. So that was the thing. So codeine isn't, they're all addictive. And I think what they use is very often when you have teeth pills, they give you codeine and Tylenol. And that's a synergistic effect kind of thing, which will help kind of thing. Or So Thebaine. So this is, all of these are in the mixture of the opioid poppy. So, I'm giving the answer. Okay, so back in the 1800s, they had a semi-synthetic, how would we make this synthetic opioid? This is heroin over here. So how would we make that?
Wait, this one is heroin?
This is heroin.
Oh, God.
This is what
You usually make it in the woods, so nobody catches it.
No, wrong answer.
Wait. You might be speaking from experience. We don't know. But yeah, so you're just talking.
Why do you think I'm so happy today? Why my leg doesn't bother me?
We're just talking about morphine on the left and derivatizing it.
Yes, that's what you do.
So what do you all see that you?
And you did this. I'm sure you did this in organic,
Yeah. Yeah, that's an acetyl group.
right?
Yeah, any
things you made acetyl group?
acetyl chloride is like one of those labs they run in organic chemistry where you're like acetylate ferrocene or something.
Or you never made aspirin?
Yeah. You use acetyl chloride with like methyl.
My problem is I taught organic for,
Salicylic acid.
yeah, from salicylic acid.
So you get some morphine and acetylate it, now you have heroin.
Right. And when that came out, it's going to be non-addictive. People, when they made a new one, so acetic anhydride, and is the reaction mechanism just in case, because we want to, but okay, and the nutsiness about that stinking compound is, remember we talked about prodrugs? Okay, so remember, so who you went to the other one, you saw what they did with Prontosil? Remember it was a little different side chain. But what happens is so what they do is they put that on, and so what happens is this gets into the liver, goes into your bloodstream, especially it goes in the first time, goes through the body, and then everything, anytime anything goes into your stomach, or it passes through the liver. So in the liver, that thing, the acetyl group falls off, and then it goes back through. So that's why heroin is even a worse, it's actually more addictive, I guess, or equally addictive. Because then, so also what happened, how they did this was it was the same thing, was how they actually made aspirin. Because salicylic acid was, what they used to use was salicylic acid, it came from the bark of, I forget which tree, but it came from the bark of a tree. And it's got a phenol group, and a carboxylic acid group. So carboxylic acids, what kind of functional, carboxylic acids are acidic or basic, right? What is it? Carboxylic acid. Okay, what about a phenol? Is phenol acid or basic? Trick question. What's a phenol, acid or basic?
Yeah, it can be both depending on pH, right? But phenols are generally acidic.
Because where are phenols in the NMR?
They show up sometimes around 10.
Right, so that, and what's 10, and where does carboxylic acid show up?
Yeah, 12 to 15.
Right, so that sucker had 2 acid functions on it. So what happened is Hoffman's father had arthritis. He probably, like me, he probably was hobbling along on pain back there. And so he could not take it. So his son worked for Bayer, and that's how they got aspirin. So now we're going to go through kind of things. So notes on, now we're going to go into the actual thing about how we do drug design. So here we have, this is basically a cartoon, and I'm going to use a lot of cartoons, but then I'm going to pull up the structures to give you a better idea of what kind. Now you show why it's going to be hard to take. I'm telling the stories. All right, so we have the messenger, and then what happens is it goes into the receptor, right? What happens is, notice how it's got this, so the structure goes into that and then goes into the receptor, correct? Now notice what happened between here and here. Change shape, right? It changed shape. So that's what happens when it changed shape, it plugs in, and then after it changes shape, that's when it releases the neurotransmitter or something like that kind of. Then after it sends it out, it exits. And what happens to the receptor? It goes back to its original shape. So again, we'll use dopamine as it's easy for me to hold. So dopamine comes in, and you'll see. Also, you remember we have these, it's going to be we're going to know all those little pieces that we know about those functional groups. We're going to pay attention to what's going to happen when we get further on. So that's what the point is. Now, what happens is I'm going to steal Tim's analogy.
Well, yeah, he's not here to defend himself.
I know.
He's another guy anyway.
Another gray, no, he's bald, not gray haired. Anyway, so he actually had to give testimony because he's a forensic guy. So he had to describe a receptor to a jury and to lawyers. So you all know what a catcher's mitt, right? What happens with a catcher's mitt? So what happens, he explains that a catcher's mitt, you throw the balls to the catcher's mitt, right? So it goes into and what happens when the ball hits the catcher's mitt? It changes shape. And that's when it does the changing of the shape. That's when it sends out the neurotransmitter. Okay?
He had to describe that in front of a bunch of lawyers and stuff?
Yes, because you have to explain to them what it is.
Was this a patent trial?
No, I think, no, it was some
criminal.
It's some criminal thing, I don't remember. All right, so what happens if it's a softball? Not a softball catcher's glove, what do you call it?
If you got a baseball mitt.
You got a baseball mitt. I qualified it. This is the first time I'm saying this, guys. I'm trusting it.
Can a baseball mitt catch a softball okay?
Not very easy,
Not very well. Why?
because it's too big.
It doesn't fit in the pocket, and then what we won't give the right release of the neural. It won't send the signal, right? So it won't send the signal down the unit. So yeah, but that's why I say right now, because I have a sciatica bother me. What's happening is when it's pulling, the receptors that keeps pulling kind of thing. And what happens is when I take, say, if you took a narcotic, that's why narcotics are so good. If you take the narcotic, it blocks it, so it doesn't send the signal down the pathway. So that's why analgesics are. And the thing is, these darn opioids, they don't just affect here, they affect here and makes you feel good. So that's what the main. And suppose you had you tried to catch it with a ping pong ball. Try to do a softball with a ping pong ball. What do you think would happen if you really fired the ping pong ball?
So you got a ping pong ball shooting at a catcher's mitt. You got a baseball mitt. Would it work?
Do you think it would work? What would happen? It wouldn't even catch it. It would just shoot off.
Yeah, I was thinking like a beach ball or something.
I'm getting there, doctor. I'm trying to get there, professor. No, right, yeah, spoiler. You can see why he and I hit it off when we first met. Anyway, so if you use a volleyball, it's not even going to get in there, kind of thing. So I just think that's so agonists. So the definitions, a few agonists. So these are the agonists mimic the natural receptor. So an agonist, instead of being a receptor, would be called instead of using an agonist, what would be another name for an agonist? It's an easy question. It's an easy question. Don't think so hard. What would be an example of an agonist? Any kind of agonist.
A fly.
What?
A fly.
A fly. No. A drug would be an example of an agonist, right? Because it mimics they bind quickly. So see, now you understand with that analogy. If I just read this agonist or some easy bind and leave, number of binding interactions, now you can understand. You understand what agonists bind and leave, right? Number of binding interactions. Why are numbering binding interactions in and out important? The number.
Every time it binds, it's going to be releasing.
Right. Or or then we'll get to the next one.
It causes some type
It
of.
causes some kind. If it's a true agonist, it's going to cause a binding and it's going to release. So it's going to have the signal's going to go in and then the nerves are going to fire. Okay. So what an antagonist would be, what? It's in the board. Is the opposite, right?
So I got a question. So when you're talking about like medicine that you're taking for your sciatica, is this an antagonist because it goes in and binds with the receptor and prevents?
It prevents when you take ibuprofen, it cuts down in the inflammation in the kinase, in the kinase thing. Be another thing. I take shots. I take shots to block the kinases for RA. So what happens is the shot goes in and stops the kinase. It stops the inflammation. So that's why aspirin, those things are in, because they block that response for, because every time you get a pain kind of thing or a fever, but you have with a fever, the fever is the antipyretics, they stop the body from having that fever.
Is it kind of like having a baseball glove that already has a baseball in it that's occupied by our antagonist?
Right, and
So
that's the thing is,
so that the natural, whatever the body wants to send to there, it's already.
it's blocked, and that's why antagonists are very interesting. When we talk about opioid antagonists, you'll understand it a little more. So, and this is the other thing, they tend to be stronger. So sometimes what happens with antagonists, they stick on another, say, side chain kind of thing. And remember, the pockets have, those pockets. What's around the pocket in the receptor? What are those receptors made out of? The pocket. No, in the pocket of what's a protein made out of?
Yeah, it's a bunch of amino acids.
Right. So what's going to be sticking out? What are amino acids? Let's go back to what an amino acid is, or what?
All these peptides. We got all.
There you got this big thing kind of thing, and you'll see them. So you got this big hole, okay? You have this or cavities, right? So the cavity goes in there, and so if you're a good antagonist, it goes in and binds exactly into the spot there, kind of thing. Sometimes with an antagonist, say it will find like a sulfur out there or another glycine or another, and maybe that part of that molecule will stick to their kind of thing. And because it has now 3 places that it holds in, it's going to bind tighter than the regular receptor. Does that make sense? Say yes. Yes? Yes, Michael?
Yes, Profi.
So, okay, so we're going, so.
Right, he goes by Profi sometimes, short for professor. So.
But no, and that's because one of my former students who was a pain in my rear before I had a pain in my rear named me that. And so when I do a STEM camp, all the little kids play, call me Profi kind of thing. And it was a STEM camp, and we were, what do we do with 7-year-olds to 15-year-olds after dinner? And so we did red light, green light. So every night, Ralphie, you promised you're gonna play red light, green light. So that's my claim to fame. All right, so here we got a binding site. These are actually these cartoons. I don't know where I can't remember where I found these things, Mike, but they were the best indications of how to explain it. And I think when you do them all through, I think it just all falls into place. So we have a binding site, an OH. We could have, there could be actually get a serine right out there. What other OHs?
We have 3-amine and
3-amine, but we get plenty of OHs out there of amino acids, and we have carboxylic. So everybody has all amino acids have a carboxylic acid, right? Oh, so what kind of interaction would this be?
yeah.
In this guy.
It's like an aromatic on
Gen Chem 1, Gen Chem 1, or Gen Chem 2.
So it's pi stacking.
Because it's like.
Yeah. Yeah. So this is probably London forces.
Yeah, but you don't even have to know that. Like, so it's going to want to, so it's going to sit.
So the kind of interaction you're looking for is likes like.
Didn't you say that? Didn't you say that? Yes. Yeah. So, okay, so here we got this, right? Right? So he's going to go in, he's going to sit in that pocket, right? And then the other guy's going to go in there. That's why this cartoon is great. And then after it gets in, it has the induced fit. So what's going on here? It's binding with the pi clouds. And what an amine and an acid, when you have an amine and a carbox and an acid, you get a salt, right? So there's going to be a nice coordination right there. So the binding site is going to go in there, right? And then what happens after it does the way? So it fits. Once it fits in there, you're going to get stronger. Notice what happened. This thing came in here, and what happened to the receptor?
So this is the baseball going into the glove.
Yeah, and this is the glove.
Yeah.
This is the glove. This is the baseball. I think that was that. I'm pleased. It's the chemist, the future chemist over there is agreeing, I think.
And the cool thing about listening to the way you teach is we can, any of us that go into teaching, we can, use it later and maybe or maybe not give you credit. That's the reason I digress. But one of the reasons I loved going to meetings was either I could steal something good, or I could go in. Especially, I went with my buddy Jim. The two, we've been doing that for 5 years. We're good. We're good. You know what I mean? Like that kind of thing. That's the point kind of thing. And because I've been teaching this a long time, I haven't seen Tim in a while kind of thing. But we talked to sometime, we were talking about, I said, oh, you got to hear his analogy. I think the baseball glove works perfect. Yeah, like in all these texts, you probably have seen in biology texts and stuff where they talk about this induced fit.
That's an induced fit.
And that's like, but then we're sitting at dinner and he says, yeah, I just use a baseball glove as a metaphor. I was like, that is an excellent metaphor.
However,
it's a lot easier to wrap my head around.
no, but see, when he said to me that when I was going over the slides last night, I thought about comparing the glove to the induced fit.
Yeah, and they talk about lock and key, and then
I think this is better. I think this is better.
I agree, and all of us who teach anything, we steal ideas from each other all the time. Sometimes not even consciously thinking about it.
Oh, yeah.
I'll hear someone say something like, "Oh, that's great. I should use that," and then I'll use it, not even remember where I got it.
Well, we have selective memory.
Yeah. That way I can't get sued.
Okay, here's your Vanderbilt. So what happens is but the structure of dopamine has been around kind of thing, and the amino acids kind of thing. So we know what the structure looks like. So how do we design an agonist? Oh, time-wise, I think I'm not doing too badly.
Oh, yeah.
Oh, wait a minute, the clock's up there, stupid. So here,
Still got a while.
this is a hypothetical transmitter, right? Van der Waals forces, hydrogen bonding, and this. All those stupid things all those things we told you about when you win. When am I going to use this? Well, this is how you're going to use it. So here, look at how this works. Possible would be, so instead of having an hydroxy, why are we sticking amine there? It still does the hydrogen bonding, right?
So these are proposed structures. that you're-
This is something how I would approach this problem as a medicinal chemist.
Right, you could try like
The
let's
old
make
way.
each of these and
The
see,
old way,
yeah, and see if they'll
before I could model it on a computer. Or look, this is a big thing of tying up and ring and make it. And what happens is because we have that rigid structure, it tends to help with the induced in the induced fit. It pops it back into that side. Or we could just have an sp2, a double, just one double bond up there. So that's how so those are the things compared to binding groups. And what we call is bioisosteres, what they can. So again, we go to the wonderful periodic table, right? Kind of thing. So think about if I have an oxygen in my molecule, what do you think would be a good thing to put in place of an oxygen that would be similar?
Yeah, just yell stuff out, guys. What do you think?
What?
Nitrogen.
Oxygen?
Yeah, someone said a nitrogen.
Why not nitrogen, somebody?
Yeah, nitrogen. Yeah,
Why not
nitrogen's right next to
nitrogen?
similarly electronegative. You know? Good job, Avery, running that camera, by the way.
Poor man.
Falling you all over the place. Yeah. Could you use the sulfur?
Why would we use sulfur?
It has the same number of valence electrons.
It does, and it also it has a what? It'll hydrogen. It acts like that kind of thing. But what's the difference between him and him?
You want disulfide bonding.
Hmm?
Yeah.
What?
You gotta say it louder, yell it out.
I'm old. I
For
can't.
some reason, you wanted disulfide bridges or bonding.
No, that would be something, but it's you guys into graduate work. There's something even
Sulfurs are bigger than oxygen.
sulfur or bigger. So what do you think would happen in our little? Suppose we stuck a sulfur here instead of the oxygen. What do you think might happen?
It's much bigger than
What do you think might happen if we tried to go into the receptor?
It's just not fit.
Or it could bind more. So we might have an antagonist.
How would you find out?
How would you find out?
Yeah, what would you do?
Test it.
Yeah, you'd have to- so
You'll make it and throw it into the assay. And if you run into the assay, it either binds, they can do the binding. Does it bind and release? So you can text binding. You always do binding first. And then they isolate the enzyme and throw the enzyme in with the drug. So initially you just do the bind, So that's how they check and they see how many go, how many they assay it.
Yeah, I want to ask you a question actually.
Go ahead.
Selenium, of course, is beneath sulfur. Would you ever use a selenium?
Never in my life. would I
And,
do
and why
selenium.
not? And I
Stinky.
suspect the answer is no, but I'm just
Yeah,
curious.
why? Why do you think as a process and as a chemist that had to live with it?
Well, yeah, I don't know. I've never.
Also, remember as you go down the periodic table, what happens to toxicity?
Yeah, so it probably gets more toxic, right?
Have you ever used selenium?
I think I don't, not that I can remember.
Okay, so here in you would have.
Yeah, and I would remember.
I
seeing
mean,
it if I did.
sulfur is bad, but selenium,
Yeah, so sulfur of course smells like diarrhea, and so
rotten
I do a
eggs,
ton of sulfur work and no
Rotten
one wanted
eggs.
to be near me. So.
rotten eggs. I find.
Yeah,
I can't, to this day, I can't stand rotten. So again, here's another agonist. I'm gonna hang myself. So here, but again, that's still lipophilic. It might, but it won't bind as tightly. But it's not going to, it may not, it binds there and it binds there again. But it has a weaker, because this cyclopropyl, there are two things that happen. First of all, it's not flat. So if you have the flat thing, it's going to sit, oh, you know, one aromatic, these two aromatics. Oh, I'm gonna this way. Oh, I'm gonna come into that kind of, oh yeah. I'm gonna bind to their kind of thing. But I don't have, but if I only have one cyclopropyl, I'm not gonna bind, I'm not gonna have interactions with all. So this is gonna be a, but so you're going to want to have that aromatic. And what may happen to, suppose here has a type tyrosine. So if you have a tyrosine over here, it may bond better because it's got more points to attach. Question? Shout out, is anyone having from the other sites? Because I don't want to.
yeah. If anyone has questions from any of the non-Logan sites, just start yelling.
This guy only has 2 interactions. They have to be, though, if you stick this OH over there, it's not going to see that OH, so it's not going to bind. And you see that in your activity. What are we going to talk about here? Okay, you all know about thalidomide, right?
Oh yeah, thalidomide. Yeah, for those who took organic from me, remember thalidomide?
So,
It causes birth defects, but also
so we have a binding site for this guy, right? The mirror. Again, 3 interactions, 2 interactions.
oh yeah, and all we have is that enantiomer.
But the enantiomer, so that's what happened was the other enantiomer, and very often you will find that with drugs, one enantiomer is more active than the other. So they both will interact at the receptor, but the preferred structure, just like the preferred side chain to quit the trovin. That's probably why he's not feeling good. I insulted his compound yesterday. But actually, his compound was the first one that was active, not mine.
He's not with us, but he's in this hotel room right now.
He's under the
He's
weather.
ill, so.
So anyway, so that explains. So we can explain how compounds react just by that. So I'm going to go that. So agonist, we said all that. Yep. Steric, you can have a steric block with them, so it won't go into the receptor. This is a neurotransmitter, the same thing. The binding groups. So, okay, so here, what we have is a bunch of selective compounds. And you don't have to memorize all these things, but here we have dopamine up at the top. We have dopamine up here, serotonin and adrenaline. These are agonists. That's a dopamine agonist. But again, look at this. There's a serotonin agonist. This has got a chiral methyl. This is probably a trick. A lot of when you put fluorines in places of methyls because they're small. They can replace, you can replace hydrogen. Butyrol is a beta-2 agonist. It stops it dilates the heart, the lungs. Again, that's chiral. Clonidine is acetylcholine. But they all have, but if you look at it, they all have a similar motif. Oh, okay, so what we have heard of, dopamine is an agonist on its own receptor. But if you go and remember, look at the structures, go back and look at the structures of dopamine and all the other ones, it also, depending on the concentration, it can react at all the other, the alpha, beta, D1 and D2, it can react at all the other, all the other receptors. And the opioid receptors are, here we go, hydrocodone. Remember I was talking about hydrocodone, oxycodone? They're what do you call it? They sure look like
Yeah.
I wouldn't be surprised that they have the same effect and the same.
Yeah, do y'all see morphine in that like upper left? It's kind of right there.
Oh, yeah.
Look at all of these other things and you'll notice that the structures are so similar. Like you see oxycodone right there in the middle. What's the difference between it and morphine? It's got an OH right there, hence the name oxy. And you see it has one at the end of it? One is a ketone, which is a carbon oxygen double bond. You see a ketone in oxycodone that ain't in morphine? You see it right there on the bottom left quadrant. See that? This is where kind of the names are coming from, right? One thing that you'll notice, by the way, is a lot of amines end in the letters INE, including morphine. Does it have an amine in it?
Okay.
It's got on that ring that's jutting out, right? The same thing is also true of cocaine, which I don't think he's got a picture of.
It's coming.
Yeah, it's coming. So cocaine, if you look at the spelling, it's got an INE.
And cocaine is a dopaminergic.
Yeah, and it's got an INE at the end of it. It has an amine. in it.
Okay, so this is what happens when I have heroin and I have fentanyl. I'm having so much trouble with names, and I think that's because my age is getting. So you see how these fit like this kind of thing. So they sort of fit. There's my oxygen, even though it's a carbonyl. You can still see the receptor, and I think the reason this guy, I think fentanyl is more active. I think because it's so flexible. When it gets into the glove, it goes, the glove comes around and can come around better. And then Mike, can you put dopamine on top of that and pass that back?
Yeah.
I'll just
pass that around. So you want me to put dopamine?
Put them, one on the bottom and one on the top,
I
sort
don't know
of.
if I'm gonna do this the way you want, but okay. So you want me to sandwich dopamine on top?
Yeah.
Like that?
And they also, you see,
You want me to have all three of these on top?
yeah, that's the idea.
No, pass them around. We're kind of saying I don't.
And
know
you may have to manipulate it a little kind of thing.
And I don't know if I've got them like lined up the proper way.
Everybody,
I look at this-
this is for him not taping my thing last.
Yeah, this is my punishment that I have to. So
Anyway.
hang on. So right here, this is dopamine, and I can see the structure really clearly. And then over here,
That's morphine.
This one's morphine. Okay. So that nitrogen.
And it's broke, be
The
careful.
nitrogen is your blue. Yeah, that's okay. I'd save it. Is the blue atom.
Okay.
Oh, can you go back to that previous slide? That was so cool. The one with the codone. So I got hydrocodone. Okay, no. So you see the oxycodone right there? Jump over to hydrocodone to its right. What's it got? It's got a hydrogen instead of the hydroxyl group.
But
Hence
what
the
do you
hydro-
think's gonna happen?
Hence
to
the
that?
hydrocodone. It's got a hydrogen. See that? Yeah, oxycodone has an oxy.
And it has, what's the difference? It still has an
And then
oxygen.
heroin has that acetyl group. So heroin looks like morphine, except it just has an acetyl group, which is a carbon oxygen double bond flanked by a methyl dangling off of that bottom left quadrant OH. You see that? And all these things just are very, very powerful narcotics. And the specific activities will be a little bit different. And all it is just subtle differences in the structures, but they often make big differences, but sometimes not. But all of them are highly addictive. This is why when I talk about Lortab, like I was prescribed Lortab when I had
Your knee?
like years ago, and I took like because I had like intense pain because I had gallstones. So I'll talk about this later. I don't have a gallbladder. I had to have it removed. So
Me too.
I'm a gallbladder.
We're not supposed to be. We're not fertile, fat, and 40.
But anyway, so the point is, they prescribed me Lortab,
Oh, that's it.
and when like they said, like here's the amount that the max amount. I knowing how addictive these things are, I took like 1/4 of the allowable dosage, and I strongly recommend it if you can get away with not taking any of them. You know, like I would just take ibuprofen.
But it does. It does. When you have, I can, you can. I mean, you can see why when people are in the pain. It does.
Yeah.
You don't think
think straight.
it's really. I mean,
And sometimes there's no way around it. Like, but my pain.
well, like
is
cancer.
manageable now if I can
Like
just get
cancer.
myself-
I love cancer. People are terminal. Oh, I don't want to become addicted. No, let the poor person have as much opioids as they want.
Yeah.
So,
sometimes there's
Let
no
me
way
know.
around
So
it.
anyway, what we do is we have opioids have different, what they call different. That's all right. Do the best you can. They have what they call different types of receptors, and what happens is these are in different parts. Like there's the mu opioid receptors. Some are in the CNS. Some are in the CNS in the peripheral. You don't have to know all this, but there's a lot more information. There's a lot more information in this that I'm trying to get to, kind of thing. And the Z opioid. So quickly, a design of the antagonist. You already know this, so not the binding site again. No change in the shape. So it binds, but then it doesn't, it binds, so maybe it doesn't have, and that's, if you think about, a strong binding would be a fastball into that glove. If you just lob the thing into it, there's not enough, it's not going to have enough energy to get in there. So that's going to be an antagonist. So, but fails, so it doesn't change it doesn't do the change in the receptor, so it sends the signal. So that's what an antagonist does.
And the antagonists, you said they stay longer.
They stay longer, ideally.
Yeah.
They have what they call mixed agonists, antagonists. I mean, it's, yeah, so the thing with antagonists, they have extra binding sites. So here again, our hypothetical, and here, remember I was talking about the pocket? So there's two other nice little areas that you can bind to. And what happens is, by changing parts of the molecule, you're going to find where those things go in. There's going to be a hydrophobic, an anionic region, and an extra hydrophobic. So if you were an extra hydrophobic, you could stick, what kind of group would you stick in if I had an extra two? What type of structures would I put in if I need to design a molecule that had extra hydrophobic? What groups would I put in
Yeah,
there?
what do y'all think?
Hydrogen,
Yeah, say it louder.
more carbons, hydrogen.
No carbon, yeah, exactly.
No.
See, it's not that hard to be.
To be a medicinal chemist, this is cake, right?
So here's the induced fit. Yeah,
Oh, yeah, there you go. There's a bunch more carbons and hydrogens on that, right? Including an aromatic ring. Good call. Avery, was that you said that?
Oh, yeah.
when I teach my med chem class and I do neurotransmitters, I usually have only 5 to 10 people. So we have a bunch of them, and we build all the neurotransmitters and we stick them onto Harrigan, and we make the giant receptor, we call it, for.
One of the things I worry about, like if I were in the lab working that, I would
Mhm.
worry about like some of those final steps about accidentally like breathing in stuff, but you're probably suited up a lot.
On scale, we're always shooting.
Yeah, you're suited.
But what they do is, what they call is, they got depending on how potent, if you have a P4, which is a very dangerous one, what do you call it? You would be fully suited up.
Yes, you got big suits, big masks.
Yeah.
You know.
So, okay, so here, histamine, have Benadryl, and then, histamine, Allegra, again, they look at the structures. But histamines, if have those, they still have, instead, they have this, they have histamine as the aromatic. So in antihistamine, so it's another transmitter, but it's got the same motif.
Yeah, you might notice, look, so that's histamine on the upper left?
Yes.
That looks like histidine, doesn't it? A
Yeah.
little bit. You remember we talked about histidine? Or
What's
I
the
went over it-
difference?
I went over it so
What's
many
the
times, you were
difference?
sick of it probably.
What's the
But
distance?
remember how, yeah, so histidine is the amino acid that of course has the carboxylic acid, the amine group. This one has been decarboxylated. It doesn't have that COOH, that carboxylate group. But other than that, it looks very much like histidine. It's got that imidazole ring dangling over there on the side chain. Do you see what I'm kind of talking about? Yeah.
And
See,
sertraline and all these
Ah.
atropine. See here, monoamine oxidase inhibitor. What happens is in the vessel, if those neurotransmitters that had the hydroxy groups stay in there too long, they have monoamine oxidase inhibitors. So we give them a drug that prevents the monoamine oxidase inhibitor from destroying from the neurotransmitter, so it stays and keeps firing, kind of thing. Because that's what happens is the idea that these drugs, what happens is they go in there, they just keep firing. And it depletes, because it also depletes the natural. That's how addiction occurs. And sertraline, serotonin, serotonin and dopamine are my two favorite. Again, that's sertraline. These are antagonists. Opioid antagonists. Morphine is the agonist. These are all selective mu capital kappa. Notice, I mean, you're not going to memorize every structure, but if you, from now on, if you ever see this structure, oh, that's an opioid. Inverse agonists are pretty neat. Pretty neat, but they are and they aren't. Inverse agonist, it goes in, and then it's got this alkali. Remember alkylating agents? Goes in here, and what happens is instead of being able to go away, it locks itself in there. And what happens if it's in there all the time?
Yeah, just stuck.
It's stuck in there, and what do you think happens? It just keeps firing.
Is this, so this looks, well, at least the way you've drawn it, looks like an SN2 magnet.
Exactly. And it binds irreversible to
Are
the
these
receptor.
also called suicide inhibitors?
Yes, I
And the
think.
reason they're called that is because the drug itself basically it goes in and doesn't come out.
It does, because you get a nice covalent bond.
Yeah, because it's covalent.
And this is a nitrogen mustard. You hear about nitrogen mustards? That's why they are. See? That's why they're so.
Are you talking about mustard gas?
Yeah, mustard gas and things like that. This is a nitrogen. These are they had nitrogen mustards, but then they decided, oh, let's make some better ones.
What's better than mustard gas?
Double much better. That's what it also has in this thing, probably sits in the receptor, so there's more binding. So it go up, it goes and go, it's gonna find, oh, I'm gonna go into that pocket, darn it. And it's gonna really hold there, and then the alkylation is gonna come in and
what does
kill.
it do?
It's gonna kill the receptor.
Does it kill people slowly, like mustard?
Well,
gas?
no, mustard gas goes all over kind of thing. And like curare is acetylcholine esterase. Stops acetylcholine from being, the acetylcholine just sits in the cleft too. That's
I'm
how
just wondering, what clinical good does this molecule have?
these are the ones though they use for, this is what they used to use for cancer initially.
Gotcha. Modify the structure.
Irreversible, so naloxone isn't irreversible. That's why it's so good. Because once it gets in there, and again, you see that once it gets in there, it's got that nice little, so it's got those two nice little alkylating groups. So once it goes in there, again, yes. But the thing is, that's why Narcan is so fantastic, because it's got a better affinity for that receptor. So it pushes out the opioid.
Yeah, and Narcan is that drug that they'll inject into someone who's overdosed, I think.
Mhm.
Am I right on that?
Yep.
Yeah.
And the last one, I think I'm doing pretty good time-wise. An allosteric. Oh, good, we'll have time for questions. So an allosteric is what happens is, again, what it does is it finds another site on the, so instead of the pocket that we want to go, it finds another nice pocket to go into. And so what it does is it it perturbs the receptor, so it doesn't go in. So the natural endogenous drug or endogenous amine doesn't go in there. It changes the shape. So I don't know, who's got an idea for an analogy for that baseball glove?
Yeah, so if you had like another place on the baseball glove outside the pocket, that something could stick and it would change the shape of the glove so it no longer could
Yeah.
catch a baseball. That's kind of what happens
Right.
sometimes.
And this is another one, the umbrella effect. You got some big that has a very, so we think it says, so it has a binding site that's close by, but then it covers the real, it's another type of allosteric receptor. And here goes,
It's blocking the hole basically.
yep. And partial agonist acts as it, but produces a weaker effect. Just opens it a little instead of opening it all the way. I think these are how local anesthetics work. But don't quote me. Oh, don't. So these exams are very high if it's partial. These are, but the buprenorphine, that's a partial agonist. So, oh, they developed this. So maybe this is going to be non-addictive. They all are addictive. Provided they get. That's the thing. If they get into the CNS, you're dead. I mean, in having activity. That's why antihistamines, the antihistamines, the initial Benadryl, if you take Benadryl, it puts you to sleep because it goes into both the brain, it goes into the brain, it suppresses the histamine in the brain and in the peripheral. So the later ones only affect the peripheral. That's why Allegra and all those are really. And another example is also you have antihistamines, you have H1 and H2. H1 is the antihistamines, H2 is in the gut. That's where people with ulcers, it just hacks that. So it suppresses the stomach acid. So that's why those work. And that's the thing you got to remember there. Receptors, they'll say beta 1, beta 2, alpha 1, D1, D2 kind of thing. So the thing is, doesn't matter. So if you're in the medicinal chemistry, we're going to look for a new thing. We found, by the time you guys are my age, there'll probably be 100 different D1 or D2 receptors. But that's and there's inverse, so these are, so I'm just showing structures here. Desensitization is very different. Over the course of time, the receptor gets oversaturated and it doesn't respond. And especially because the agonists are more potent sometimes, it doesn't respond to the natural one. That's how addiction occurs. I did it.
Well, everyone, let's give a round of applause. Thank you. And by the way, what happens at 6 minutes when this class ends, everyone gets cut off and we can't stop it.
Oh, okay.
So it's a hard
Well,
act.
does anyone outside, out in the,
Yeah, the non-Logan sites, anyone have any questions out there you want to ask? Looks like someone's having a hard time.
he's tired. I wore him out. Nobody? What?
What, any questions?
Oh, what about here? Sheldon. He's not listening. Oh, you guys are mean. Okay, who's got a question? Come on. Yes.
So does each body system have its own unique receptors that are specific to that system, or are they
Each
all,
body? What do you mean? Each
Like-
person?
each system within the body?
They all have different receptors. The dopamine, histamine is they're all in different parts. That's why you saw all the opioid receptors. They're in different. The neurotransmitter, the endorphins work on all of those areas, but because they have the mu, the kappa, they're in different areas and by tweaking the structure, you can have it go into the peripheral or to the brain or both kind of things. That's why the opioids are so horrible. They affect everything. They affect the gut, they affect your breathing, they affect everything. So that's why they're so.
I mean, it sounds like you're asking, like, does like my nervous system have different receptors than my
Yeah,
digestive system?
Yes, but they all have like, all of them have opioid receptors. They're just different, they're different shapes.
So opioids will go into everything.
Right, because the neurotransmitter is, and the histamine is a neurotransmitter of the stomach and for the brain kind of thing, for putting you to sleep kind of thing. So what happens is, if it doesn't get into the brain, it's not going to put you to sleep, but it'll help, but it'll help that kind of thing. Yes.
They're kind of like what Dayquil and NyQuil are, is that?
Dayquil and NyQuil are antihistamines.
Yeah, and so is Dayquil like it doesn't attach in the brain?
It doesn't, yeah, it has a initially, what happened to them, why they had, all of this came out with the histamines kind of thing, how we got CNS drugs to treat some of these horrible disorders like mania and things like that. What happened in the 30s and 40s, they had made antihistamines. And what happened is people had rhinitis, seasonal rhinitis, and they started giving it to these patients, and they noticed that was a big difference in that. If you ever know someone that has any of these things kind of thing, they tend to make people sleepy. So the newer ones, they tried to stop to have the peripheral, like they'll have Parkinsonian because it's in the peripheral. So, but that's how they initially found out that's how they initially made drugs to treat all these CNS problems. Yep.
So to make a drug last longer, do you need to like make the attachment into the receptor stronger, or is it a different mechanism?
It's usually a different mechanism. It usually has to do more. See what happened. I mean, were you at my Trovan talk? Okay. What happens is, do you remember what about what was the great thing? One of the best things about Trovan. What was one of the things we needed to have?
The half-life was 9 hours.
Right. So what the idea is, just because that's my beef about everything being in vitro. The half-life is very important, is actually how you control that. And that you can tweak the drug so it doesn't do the metabolism. So you just can't just worry about how it looks at the receptor. You have to say, what happens when it goes through the stomach? That's why you can't give some drugs orally. They have to be IV. Or why giving something IV is so much better. Anyone?
Any last questions, students? Oh, yeah.
On a chemical level, what causes a neurotransmitter or a drug to be ejected out of these receptor sites?
It's just the way it is. It's called a turnover rate. It just it goes in, does its thing, and then what happens is it releases. I mean, just like it released the glove. So it goes in there, sends the signal, and then it reads back, and then another guy comes in.
Are there instances when like a mutation can occur?
Oh, yeah. Yeah, that's some people. So I think that's what's happening. There are some people that can't take some Asian people don't have enough alcohol dehydrogenase, or Indian people, they don't have enough alcohol dehydrogenase in their system. So that's why they tend to have more trouble with, right. Yes.
Yeah, one of the things I was going to say on that release, part of it is because of the intermolecular force. The forces that are keeping them in are intermolecular forces, which are way weaker. You remember me talking earlier. I talked about how hydrogen bonding, although it's the